Good evening, everyone. Welcome to the third webinar in the Fall 2020 series presented by Newton Conservators. My name's Beth Wilkinson, and I'll be the moderator of this virtual event. Barbara Bates is our wonderful technical director. As most of you know, Newton Conservators is an all-volunteer nonprofit established way back in 1961 that connects people to open space, that works to preserve and to maintain open space in Newton. All of us on the Newton Conservators board are grateful for the beauty and sustenance that the land provides for humans and the other creatures with whom we share the earth. We acknowledge the Massachusetts stewardship of this land that kept its ecological communities vibrant, strong, and interconnected for thousands of years, and hope to work with them as we strive to maintain and restore our open spaces. Tonight's presentation is by Matt Pelikan, the director of the Martha's Vineyard Atlas of Light project at BiodiversityWorks. Matt will teach us about Orthoptera, grasshoppers, crickets, and Katydids, and explain why they deserve far more of our attention than we get. Although at this time of year, they do get our attention late at night. We're very fortunate that Matt is here to share his knowledge and wonderful photos with us. Welcome, Matt.
Thank you for that very nice introduction, and thanks to everyone for joining this evening. Orthoptera is kind of a non-starter as a topic. I'm not really sure why, but part of my job tonight is to persuade you that you should be interested in these little critters. I'm a vineyard resident now. I've been down here for about 26 years, but I grew up in Lexington, Massachusetts, so I know the Newton area pretty well. I know you've got some wonderful conservation properties up there. And I hope that some of you will be inspired to go and chase down some grasshoppers there. I'm going to start sharing my screen, and I'm going to remember how to turn on share sound.
We're all set.
Alrighty. So the title of this evening's presentation is Springers and Singers, for obvious reasons. I'm going to try to accomplish a couple of things this evening. Partly I want to just introduce you to Orthoptera because, as I said, they're really not well known by the general public, even though they're a very diverse group and a very interesting one. I'd also like to talk a little bit about the Orthoptera, specifically of Martha's Vineyard, even though you don't live there. Maybe some of you visit, but the impetus behind this presentation is about 14 years of field work that I've spent studying these Orthoptera down here, so I'd like to pass on some of my findings. Third purpose is to introduce some important resources for amateur nature study, and the conservators will be sending around the material from the final slide of my presentation, which includes some websites and books and things that you will find useful. In particular, I commend to your attention the websites, community science websites like iNaturalist and BugGuide, which some of you may already be using. And if we have time, I'll give you a little introduction to them at the end of my talk. But the main thing is that I hope I inspire you to start doing the same kind of thing that I spend my spare time doing, which is walking around photographing bugs and trying to figure out what they're up to with their spare time. When I became interested in insects, well, about 30 years ago, I'm a longtime birder who's gone over to the dark side. Things were very, very different. I was photographing insects with Kodachrome 64 in a manual camera, and I would shoot $30 worth of film, and I might get 1 or 2 pictures. The web didn't exist then, so all of the technology resources, digital cameras, the internet, email, really were either nascent or didn't exist at all. So things have really changed markedly since then, and life has gotten just a heck of a lot easier. Anybody can go out today and get pictures of a bird that would have been to die for 30 or 40 years ago. So it's now possible to access information and through community science platforms and social media websites. As absurd as Facebook is and as destructive as it can be, it actually is a great place to learn about bugs. There are all kinds of groups that are dedicated to natural history study. these resources can bring you into contact with literally with world-class experts. You can post a bug in a Facebook group, and 10 minutes later, some world-class expert in Madrid or who knows where will tell you what it is. It's just amazing how much information is available and how accessible it is. And the study of insects and other wildlife has never been more important than it is now. We're in an area of habitat fragmentation and climate change. Species are on the move, species are endangered. So maintaining a good snapshot, a current snapshot of what occurs where and how these species are doing is really important. And amateurs can help with that. And finally, the main reason, I guess, for studying nature is because it's fun. You can satisfy your own curiosity and you can get a great deal of enjoyment out of studying insects. And I talk about insects, I mean natural history in general. My story starts with, in terms of Orthoptera, in February of 2010, when I was poking around outside on a mild day, planning my blueberry pruning campaign for the spring. And as I poked around the blueberry bushes at the south end of our house, I saw grasshoppers hopping around. There were green ones and there were brown ones that looked like this guy on the screen. Excuse me. And I was just blown away. It's like it's the middle of February. What are grasshoppers doing hopping around? And I realized that I knew nothing about Orthoptera. I could identify one grasshopper. I knew the Carolina grasshopper. I knew two crickets, and I knew that Katydids were green, and that was really the extent of my knowledge. So I had no idea what I was seeing, and I had to find the answer to that, and the result was a real commitment to study Orthoptera. I watched the grasshoppers in my yard get older and develop, and eventually they turned into something looking like this, at which point I was able to identify them as a grasshopper called Cortophaga viridifasciata, a green-striped grasshopper. The females of this species are usually green like this. Males are usually brown. They also come in other colors, but I'll talk a little bit more about that later on. And one unusual thing about Cortophaga viridifasciata is that it overwinters as partially grown nymphs. Grasshoppers and other Orthoptera have what's called partial metamorphosis, and they come out of the egg looking pretty much like adults, but much smaller. And they go through a series of molts, usually about 6 molts, until they reach maturity, gaining longer wings and larger size as they progress. And most Orthoptera overwinter in egg form at our latitude. There are only a couple of grasshoppers that overwinter as nymphs. So that was what I had come across, was these partially grown grasshoppers that are never really fully dormant in the winter. They come out and they bop around on mild days and even continue their development during the winter. And then they mature in August and May and lay eggs, and the cycle starts again. So I mentioned a couple of these characteristics already. The Orthoptera generally are well adapted for jumping. There generally is one generation per year. They take a long time to develop, and so the length of the growing season is really a determining factor ecologically for where an orthopteran species can occur. A lot of them, if you look at their distribution of records, there's a very, very clear latitudinal or elevational cutoff of where they can persist. Many species, not all, but some species have the capacity to produce sound, either by scraping body parts together or by whacking their wings together in flight. And they're mostly vegetarian, but you do see them eating, either scavenging or sometimes even capturing other arthropod prey. All right. So there are some challenges to studying Orthoptera. They're fairly large insects, which is good, and they make noise, which makes it easier to tell when they're around and sometimes to find them. But they're really good at concealing themselves, and they often have cryptic coloration and behavior. So studying them is really, really difficult. Moreover, a lot of species that are closely related to each other look very, very similar, and you can't always tell what you're looking at unless you can get a specimen in hand and look at it up close. And within species, there is often a lot of variation. So you may think that you're seeing multiple species when in fact you're just seeing one. People's Exhibit A is Orthoptera pallidna. It's a so-called stridulating slant-faced grasshopper. And these are all the same species. In fact, they were all taken within about 50 feet of each other and over the space of about 10 minutes. So they really were occurring in the same setting. You can see that in spite of the varied color, there are some features that are consistent. The sloping face, the wing length stays about the same. And on this part of the body, there's the so-called the pronotum. There is a little black triangle and a sort of hourglass-shaped white outline. And those are, in fact, helpful field marks, but the overall color of these grasshoppers is useless for identification. This is probably the most common and widespread grasshopper in Massachusetts, Melanopolis femorubrum. Again, enormous variation. You might think that guy in the upper right-hand corner would have to be a different species, but no, it's the same thing. Excuse me. There is also the problem of closely related species, separate species looking very much like each other. We've got 4 species here in 3 different genera. And if you saw them under field conditions, you'd be hard-pressed to tell which was which. They're fairly difficult to identify at sight. So a large part of my process of studying Orthoptera, and what your process will be if you choose to study bees or hoverflies or whatever, will be learning what characteristics are the ones that really matter, so you can get photographs of them and get a good look at them. I stuck to my guns and I kept my nose to the grindstone, and over the course of the 14 years of work, I've documented 66 species on the vineyard. There are probably, 80 some in Massachusetts, maybe a little bit more than that. Islands always have a smaller diversity than the adjacent mainland. But it's a pretty good diversity, and it's a very interesting fauna, and I'd like to introduce some of those critters to you tonight. One wonderful resource that I came across was this monograph. It's a reprint of a monograph that was published in 1920. And it is a wonderful snapshot of the status of Orthoptera in those days. And so when you do a project like mine, you can compare what Morse had to say about things with what you find, and you can find that species have changed their distribution, species have disappeared, they've become more common or less common. It's a really valuable resource. A lot of this material is available in Google Books, and some of it has been reprinted by organizations like these Select Scholar people. This is a species that didn't occur here when Morse was writing. It's the Philopalpus pulchellus. Handsome trig is one common name for it. I can play their song for you a little bit. They're sort of a metallic sounding. It always sounds to me like an exhaust system with a screw loose. They have an interesting distribution now this is the records of this species in New England from iNaturalist. And you can see that even though they didn't occur here 100 years ago, they're now pretty widespread, moving up the warm climate of the Connecticut River Valley and the Hudson River Valley, in the urban heat island, in the suburbs and areas around Boston, but they're absent from the higher elevations of the Worcester Hills and the hill towns and Berkshires in western Massachusetts. Partly this distribution also reflects where there are people, because these are where people have observed these things, but the distribution probably is not accidental in terms of where these species really occur. Note that they haven't turned up on the Vineyard or Nantucket yet. There are a few Upper Cape records, but I'm still waiting for this species. It's not through lack of trying that they haven't been found here. This is another one that Albert Morse would not have found in New England. It's not in his monograph. Jumping bush cricket, a Hepithus saltator. They have a pretty distinctive song. And like a lot of Orthoptera, you sometimes get a chorus of them calling in the same area. And it's really funny because each one will be calling on a slightly different pitch. So they're all a little bit individualized. Hepithus is another species, and this distribution is almost exactly the same as what we saw with Philipalpus. Only this species has made it to Martha's Vineyard about 10 years ago and still hasn't made it to Nantucket. So things are changing with the climate. Species are moving northward, and it's important, in my mind anyway, to keep track of that. So let's look at some critters, and let me try to explain and give you a sense of what kinds of things matter when you're studying Orthoptera. This is a very common species, the Carolina grasshopper, Dicicera carolina. Well, it's a family called band-winged grasshoppers for a very good reason. Their underwings, their hind wings, are usually distinctively colored. Sometimes they're clear, sometimes they're yellow, sometimes they're black with a pale margin. This is the only species in our region that has this coloration. So if you see a grasshopper in flight and it's got a black wing with a pale margin, this is the species that you're seeing. So that's a meaningful characteristic, even though we've got a pretty generic looking grasshopper with seemingly no distinguishing characteristics. And this is a close-up of the pronotum. And you can see how it's got a sort of elevated keel on it, which a lot of, but not all, band-winged grasshoppers have. And it's got this asymmetrical notch here, a vertical edge and then a curved edge on the rear. And that also is a really distinctive feature. The pronotum geometry is a really important characteristic in Orthoptera, and this is a really good example of that. Here's another band-winged grasshopper, Encoptolophus sordidus, and you can see it also has a keel and it also has a notch, but that notch is symmetrical. And overall, this grasshopper is much more compact with shorter wings. It's an interesting species because in Morse's time, this grasshopper was common enough, so he said it sometimes was a nuisance. It would eat crops and damage economic resources. And now it's a very rare species in New England. There's only one population that I'm aware of on the vineyard, and that population is in a little patch of grassland that I know from archive of aerial photographs has been in an open condition at least since 1935. So there has probably been a continuous population of these guys in the same little, not even football field sized patch of habitat, and they're just persisting, and that may be the only place they occur on Martha's Vineyard. Some grasshoppers are generalists. Others, like Trimeratropus maritima, are habitat specialists. This occurs on dunes on the vineyard. You might find them on sandy freshwater beaches in your area. The species extends all the way out to Nebraska, I believe, but it always occurs on sandy habitat and usually near water. We find it being very, very common in dune grass habitat on the Vineyard and the Cape and Nantucket. This is another habitat specialist in the band-winged family, subfamily, Cnidia fenestralis. Again, it likes bare mineral soil. They must eat something, but I've never seen one on any kind of vegetation. They just bask in the sun on bare mineral soil most of the time. Very, very strange behavior and very good cryptic coloration. Populations evolve to have, this isn't a very good example of that. Maybe the a seaside grasshopper is a little bit better. They evolve to match their substrate, so they're as invisible as possible. This is a different subfamily. This is the Gomphocerinae, the slant-faced grasshoppers, so called because of the way that their face recedes. This one, Chloealtis conspersa, is moderately common across New England, or southern New England anyway. This is a male. Females don't have this black on the side of the pronotum. Just a very, very pretty grasshopper in my view. And some of the slant-faced grasshoppers are really bizarre, like Pseudopomma brachyptera. This was mentioned as being on Martha's Vineyard in the Morse monograph. And there were a few specimens taken in the early 1990s by some survey work that was being done by the Nature Conservancy. But I was unable to verify its presence for many, many years. And I finally found one individual in Edgartown a couple of years ago. And I think that's a really important point. It shows that grasshoppers are able to, and other Orthoptera, can persist for long periods of time at a density, a population level that is essentially not detectable. And unless you are lucky and stumble over one, you'd never know they were there. So if you can't find something, don't despair. They may still be around. It may take you have to be patient. It may take a lot of years before you've amassed enough time in the field to encounter one. The so-called spur-throated grasshoppers are another major division. I don't have a good example of the spur, but in between their front legs, they have a little tiny point, a little spike pointing down. Nobody knows what it's there for, but anatomically, it was what characterizes this group. Hespertetyx viridis brevipennis is the subspecies we've got here. It may actually be a good species because this subspecies is isolated geographically from the other subspecies, and it actually is a very rare grasshopper in southern New England right now. Here is where it occurs according to iNaturalist records. You can see there's a cluster on Martha's Vineyard, and then there are a few records from Miles Standish State Forest in Plymouth. And those are the only records in the Northeast that are in iNaturalist. So this is a specialist that you find in sort of areas that have goldenrod and blue stem and some scrubby wood like scrub oak vegetation. The main genus in the spur-throated grasshopper department is Melanoplus. There are about 240 species in North America. Some of them are easy to identify at sight, like Melanoplus differentialis. Note the yellow tibia, which is pretty unusual in grasshoppers. And note on the hind femora, these bold black chevron markings, which are pretty distinctive. And then this overall sort of putty gray color. They also show up in a little bit more of a yellowish variant, but it's a pretty distinctive grasshopper. You can recognize them at sight, no problem. Unfortunately, most Melanoplus look pretty much like this one, just sort of generic grasshoppers. They've usually got a black patch on the side of the pronotum, which may extend onto the head. They often have some diffuse banding on their hind legs. A lot of them have red tibia, but they all look alike, and it can be really, really difficult to tell what you're looking at. Fortunately, male Melanoplus have this organ in the back of their body at the tip of the abdomen called the circus, or the cerci for plural. And here is a close-up of the one on Melanoplus keeleri that we're looking at. And see this little mitten-shaped thing here. Every Melanoplus species, the males have a distinctively shaped circus. So if you can get a photograph like this, any Melanoplus male will be identifiable. You still can't identify some of the females in the field or from photography, but it's a good thing to know that this is an important part of the body to get a picture of if you think you're dealing with one of these. The pygmy grasshoppers are actually a separate family, and as their name suggests, they're very, very small, usually less than an inch long. And this is the pronotum. The pronotum is elongated on these. It extends all the way out. The female has a long one, just as an individual variation in this species. The male in this mating pair is a little bit shorter. But these aren't wings. All you can see a little bit of the wing here at the end, but all the way down to this point where my cursor is pronotum. So it's a very distinctive morphology. Katydids lateralis is a species that is going to be much more common up your way. It's known on the vineyard only from one record out in Aquina. But these are very small and difficult to see, so they could be overlooked anywhere, really. Here's another one, Tetrix ornata. And this is known on the vineyard only from a couple of individuals that I found in a commercial strawberry field. And that gave me pause. So I talked to the farmer. He said, oh yeah, those strawberries, I ordered them from a place up in central Massachusetts. They came to the vineyard as plugs, flats of plugs, and it probably came with some eggs or possibly some adults of this species. So I don't think this species occurs here, but it can be imported. That has happened a lot of times, actually. There are a lot of records of Orthoptera that have been transported long distances as far away as California, turning up on the vineyard in association with agricultural products. Let's talk about crickets a little bit. This is a very common species. It's actually part of a species pair called the fall field cricket and spring field cricket. They are separated only by seasonality. They sound the same, they look the same, and it's a very familiar call. You probably got these guys in your basement even as we speak. Very common species. I've done a little bit of work with visual analysis of sounds, and this is actually the Springfield cricket, but very similar sound. And what I've done here is I have a known individual from a website called Singing Insects of North America, and I have a sound recording that I made in my backyard, and I process these with a program available from the Cornell Lab of Ornithology called Raven that will generate this kind of visual output from sound files. And you can see these are pretty clearly the same species, or at least a very, very closely related species. The fundamental tone is at the same frequency. The calls are spaced about the same way apart. The main overtones are about the same place. I actually captured a high overtone up here at about 18 kilohertz that the reference recording didn't have. But I think there's a lot of potential here for identifying or calling Orthoptera by using their sound. That's how they sort themselves out, after all. The ground crickets are a smallish genus. There are actually a couple of genera. They're very small crickets. They're about a half an inch long in terms of their body length. You'll rarely see them. They're very difficult to get a good look at, but fortunately they all have pretty distinctive calls. This is a large ground cricket. It sounds kind of like a field cricket call, only vastly sped up. And we have about, I think, 6 species in southern New England in this genus. So they're most easily distinguished by their calls. They all look alike, and I don't think the visual identification of them has been very well worked out. The tree crickets are lots of fun. These are indeed crickets. They look a little bit more like katydids than some. This is one of the more distinctive ones in the main genus Acanthus. The blackhorn tree cricket, or Acanthus nigricornis, so called because it usually has black antennae and also black markings on the legs. This is a male, and his wings are raised to call. He rubs them together, and the rest of the wings serve sort of like a sound radio, like a loudspeaker, and they sound like this. And there are 6 or 7 members of this genus in southern New England. Some of them are distinctive, like nigricornis, but a lot of them are not. This is the 4-spotted tree cricket, Acanthus quadripunctatus. And several of our species look almost exactly like this. You really can't tell them apart visually. You can tell them apart in terms of their calls. So that's a much mellower and more musical call than the last species we looked at. But even more handy than that is the fact that all of the Acanthus species on the basal two segments of their antennae have patterns of spots that are distinctive, in fact diagnostic. So the four-spotted tree cricket is so called because each antenna has this pattern of four spots on it. So if you find a tree cricket and you can't identify it, this is the angle you want to get. I do it by getting them in the hand. Sometimes you can just get lucky and they sweep their antennae back and you can get a shot. But that's really what you need to see. The katydids are leaf-like animals. There's a lot more variety than you think there's going to be. I think there is something like 21 or 22 species that I've documented on the Vineyard, and there are another 8 or 10 that can be found elsewhere in New England. Their calls are not particularly useful, with a few exceptions. You may have heard the common true katydid, which has expanded its range in southern New England over the last couple of decades. And it has a call that goes something like cha-cha-cha, cha-cha-cha, cha-cha-cha. And you often get large numbers of them chorusing in a small area. But most of the other katydids have really kind of disappointing calls. They're often just a little or else a wispy little trill. So it's not much help, and they also mostly call at night. So it's not very useful from the observer's perspective in terms of identification. But there are ways to identify these animals pretty readily. This is a female. You can tell because she's got this organ called an ovipositor at her back end, which is used for inserting eggs into vegetation. And Scuderia furcata is distinguished by the female having a red ovipositor. At least our populations do. I'm not sure if this obtains elsewhere in North America, but at least in New England, that's a pretty reliable indicator for this species. You can compare that to the curved-tailed bush katydid, Scuderia curvicauda, which looks very, very similar, but it has a green ovipositor. There are other differences. Curvicauda, the backside of the hind tibia is usually quite reddish. Not so true on furcata. The wings are a little bit different. You can't really see the wings shape difference because of the way I'm holding that. But the tips of the wings are shaped a little bit differently. So there are other ways to tell them apart, but the genitalia, the terminal appendages on the abdomen, are really the key for identifying these katydids. It is true of the males as well. There is a piece of anatomy called the supraanal plate, or the plate that goes over the anus. And it is distinctive in the males of all of the Scuderia species. In one species, it's absent, which is pretty distinctive. But the furcata means forked, and you can see why it is called the fork-tailed bush katydid, because the males have this Y-shaped. And you can compare that to the same piece of the body on curvicauda. Looks very, very different. So sometimes you can get a shot of this or a look at it in the field. More often you need to capture the insect temporarily, hold it by the wings as I'm doing in these pictures, and manipulate it so you get a clear view. But again, these features are diagnostic, and they make it possible to identify insects that you probably wouldn't be able to identify to the species level any other way. Another katydid genus is Amblycorypha, the round-headed katydids. There are probably only 3 species in our region, but the genus was just revised recently with a whole bunch of new species split off, and the ranges of those species and the ways to tell them apart in the field have not fully been sorted out. So for all practical purposes, this genus comes down to ones that have long wings and ones that have short wings. And by long, I mean the wings are longer, significantly longer than the femurs, the femora. So that dimension versus that dimension. This is probably Amblycorypha oblongifolia, but I'm not really sure. It's a male because this is the so-called stridulatory area. They rub together to produce their call, which is a very disappointing call, I'm sorry to tell you, but there you have it. This is the short-winged Amblycorypha, rotundifolia, and you can see now that the femur here extends beyond where the end of the wing would be. So it's a much different body shape, even though they're in the same genus. And this would be a female, you can tell from the ovipositor. One cool thing about Amblycorypha, and indeed about some of the other katydid genera, is that you occasionally get individuals like this, either bright pink or bright yellow. Weird, presumably just a genetic abnormality that they have a gene for pink that the others lack, or their gene for green doesn't get expressed, or who knows what's going on. They're rare, but they're not that rare. They're probably, I think they're estimated at like 1 or 2% of the population. But it's a really startling thing to find. I would think that there is a lot of evolutionary pressure against these surviving to reproduce, although this is a fully mature female and she seems to be doing just fine. So maybe it's not as much of a liability as we thought. And a tip of the hat to my friend Bert Fisher, who photographed this out in Aquina a number of years back. All the other photographs are mine in this presentation, and most of the sound recordings are from that Singing Insects of North America website that I mentioned. I'm going to finish up my discussion of our Orthoptera with 2 videos. One is going to be this one of so-called, what's the common name? Common meadow katydid, Orchilomum vulgare. I guess I have some Conservators coming up too, so we're not quite done yet. But this is what they look like and sound like when they're singing. The volume is not so loud. This is just a cell phone video. But you can see his little wings vibrating, and you can hear his call, which is like tick, 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 bzz, tick, 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 bzz. He's calling from a stalk of little blue stem hanging upside down, which is how most of his members of his genus prefer to call. When you disturb them, they just let go and they drop down into the clump of grass at the base of the plant, and they disappear, and you're never going to find them ever, ever, ever. So you basically get one shot at finding these things. Orchilomum vulgare has an interesting distribution. I remember I was talking about how they often have a pretty fine latitudinal gradient where they stop occurring. And this is an example. Aside from one record up on the North Shore, all of the Massachusetts records are from Martha's Vineyard. We're pretty much the northernmost extension of its range along the East Coast. That's likely to change with the climate, and I expect this one probably is a artificially transported one, but if it finds another one, it's probably perfectly possible for it to, for a little satellite population to get started up there. Some of the other so-called meadow katydids fall in the genus Concephalus, which means conehead, and they all look pretty much alike, and their habits are all pretty much alike. They like a mix of herbaceous vegetation in open habitats for the most part. The way to identify them is by their genitalia. Again, females have, for example, an ovipositor In this case, a very long one, longer than the body of the insect. Kind of straight-lanced meadow katydid, I think, is the usual common name, Concephalus strictus. Very common on the vineyard, less so on the mainland. In most individuals, the wings are very, very short, but every so often you come across a female with wings that extend past the end of her abdomen. And this is an interesting phenomenon that occurs in a variety of other Orthoptera species. And apparently it's triggered by crowding. As a population gets excessively dense, a gene in some females gets triggered, which produces long wings at the end of their development. Nobody really understands the full mechanism involved, but there is a genetic component, and it is the expression of the long wing gene is triggered by overcrowding. So Concephalus strictus, the other ones, we probably have, I think, 4 members of the genus on the vineyard, all have much shorter ovipositors, but the ovipositor is the key. Concephalus brevipennis, it means short-winged meadow katydid, which is ironic because the way you identify it is that its wings are longer than most of the other species, but go figure. This is a male. Males have, again, these are the so-called cerci at the end of the abdomen, and those are distinctive in this genus. So getting a look at that, a good overhead shot like this, is a pretty good way to identify them. Wing length is also helpful, and again we can see the stridulatory area here where this male would rub his wings together to produce his rather wispy, disappointing song. He's on a sweet fern plant in front of my office, and sweet fern is a pretty good indicator of the dry grassy habitats where I find the largest numbers of them. On the mainland, they seem to be a little bit more associated with wetland edges and grassy wetlands, but they're pretty much of a habitat generalist. This one is not a habitat generalist, Concephalus spartanii. It is the salt marsh meadow katydid. It's very closely related to brevipennis, but it is an obligate of Spartina salt marsh, where it is adopted to a saline environment and periodic inundation. This is a female, again, with an ovipositor that looks very much like the one on brevipennis, except it's a little bit more curved. So you're not going to find these in Newton, but if you go to the coast anytime in the late summer and you have an opportunity to stomp around on a salt marsh and you notice little brown or green grasshopper things hopping around, this is probably the species that you're looking at. They can be mind-blowingly common in salt marsh, dozens of them per square meter sometimes. So it's a very, very common species. It just has a very specific habitat requirement. And the final genus we'll look at today is Neoconcephalus, the new conehead. And the genus is called that because every species has this extension on the front of its head. And that cone varies from species to species in how long it is and how it's shaped and what the coloration is. There are often black and white areas on it. So a head-on view or a view looking up into the face from underneath the insect is really helpful for identifying them. This is the round-tipped conehead, Concephalus retussus, and it's so named because its cone is almost a perfect half circle at the end. This is a female, very, very long ovipositor on this species. Ovipositor length is another useful characteristic for identifying them. But the key really is getting a good look at the cone from all possible angles. You can also identify them by sound pretty readily. This species has a sort of wiry buzz that comes from herbaceous vegetation. They will also call from bunchgrass clumps like Orchelamum will. This one was, I think this was in my front yard. The recording was not, but the individual was. So pretty recognizable call, but it's not nearly as recognizable as the call of the robust conehead, Neoconchocephalus robustus. And this is one calling from a big blue stem patch in my yard, and they have an unmistakable call. It is like having your teeth drilled. We were sitting watching TV one night, and this guy fired up in the front yard 30, 40 feet away. And my wife had to close the windows and turn up the volume so she could keep watching her show, while I, of course, went out with my cell phone and a flashlight to try to get a video of it. That's just how it goes in our family. Very distinctive call. This is a species that occurs on like weedy roadsides and in areas with bunch grass. But interestingly, on the vineyard, it also occurs in dune grass habitat along the shoreline where there are patches of Ammophila dune grass, just like you would find the seaside grasshopper in. I don't know. It's hard to explain why species would have preference for two habitats that were so different from each other, but there you have it, and I'm sure that these guys undoubtedly know what they're doing. So some resources. I've mentioned BugGuide, and I've mentioned iNaturalist. I will try to, I think I have enough time to do a very quick look at iNaturalist. These are really important resources for studying insects, and you will be receiving an email in a day or two that will include all of this information. I'll call your attention to a website that I administer as part of my program on the vineyard that has a lot of information about vineyard biodiversity, including checklists for a bunch of different kinds of wildlife. So this information will reach you, and you are able to will be able to check these resources out for yourself. All right, so this is what you get when you land on iNaturalist. It is the, sort of your feed, as it were. You could say that iNaturalist is a huge database. It's got about 7 million people participating worldwide. It has something like 160 million observations, and it's got representations of about 400,000, 430,000 species worldwide. So it's a huge, huge, huge database that you can contribute to. Like any database, the data are searchable. You can filter them in various ways. And when you have an account and you log into it, this is what you get. And let me first go through the process of what adding an observation consists of to show you how that goes. You click Add Observations and you get to a screen like this. And you can just drag and drop. I'm just going to pick a photograph pretty much at random here. And let's see what iNat does with it. So if your photograph has a date and time stamp on it, because your camera records that or your phone can record it, you can use iNaturalist directly from your phone using an app. It will appear here. If you have a camera that will record the location, the coordinates would appear here automatically. And when you put your cursor into the species name field here, it offers a whole bunch of suggestions. In this case, it's way off base. Usually it does a better job than this. This is actually an Andrena bee. It's Andrena calmii. But if I went with Anthophila, it would at least be correct. Let me do a little bit to get rid of that observation and maybe pick one that iNaturalist will be able to do a better job with. Here we go. So Atlantic Bass Scallop, there we go, that it identified it for. So there is an artificial intelligence utility that will visually match your photograph as well as it can. And once you've got an observation in iNaturalist, it becomes visible to everybody else on the platform, and human observers can then confirm your identification or provide one if you haven't been able to come up with one. I confirmed the identification earlier of Cortophaga vertifasciata, a nymph getting ready to overwinter from up in your area, it looks like. So it's possible to comment. It's possible to tag people. So if you know an expert, you can tag them and have them weigh in on it. Everybody in iNaturalist has a profile like this one. You can see that I'm following a bunch of people and a bunch of people are following me. That means that my observations turn up in their feed, and in this case, their observations turn up in my feed. You can also customize iNaturalist so that you subscribe to various things. So mine is set up so that all Orthoptera observations from Massachusetts turn up in my feed. And all the bees, which are the thing that I'm working on now most, from Martha's Vineyard turn up in my feed. So you can customize what material you see, and you can you can message people offline like you can in Facebook. So it combines the merits of a natural history database with the advantages of social media. And it doesn't have any of the disadvantages of something like Facebook. There are no ads. They don't steal your data. It's a very, as far as I can tell, a very benign utility. So let's see, I'm going to look very, very quickly at one other thing, and then I will turn it back over to the moderators. I want to show you the BugGuide website, which again is a huge database of arthropods of all different kinds. And they're basically arranged taxonomically. So if we go to the flies and click Browse, here are the main taxonomic subdivisions of the flies. And if we want to narrow it down, you can go into here the Muscoidea, superfamilies. We can click on that, and it's working slowly today, so it may not help us out very much. But eventually, the families within this superfamily will show up here. And what I often do, you can click on a picture or on a taxon. It's not cooperating at all for me right now. It would take you to a taxon page that would show you, here we go. That would show you an opportunity to get information on the species. It will show you an archive of all the observations of it. So what I often do is have this open at the same time that iNaturalist is open. And if I don't know what something is, I will go to and use the suggestion that iNaturalist makes, and I will go to BugGuide, and I'll just put into my web browser in a new window, search BugGuide for this taxon name, and then I'll be able to see examples of the picture and compare it to what I photographed. So there are lots and lots and lots of resources out there, some of them based on artificial intelligence, some of them based on human intelligence, but it is now possible to, for amateurs, you don't need a dichotomous key, you don't need to be a scientist to identify these things, you don't need to collect specimens, although you sometimes do. There are still some questions that you can't answer from photographs. But this is truly a golden age for nature study of all kinds, with technological advances and advances in knowledge that have just made life incredibly, incredibly easy. So I'm going to stop sharing at this point and invite the moderators to chime in with any questions that have come up.
Oh, terrific. Thank you.
Whatever tricks you want.
Great. Thank you, Matt. Thank you for such incredibly detailed information for us. Thank you for amazing photographs. Just thinking about slant faces versus cone heads. I mean, it's great. I now have a sense of that. Now I've got to learn where to find them, though. And what I want from you is a Merlin for Orthoptera. So when you have spare time, that would be really wonderful. So
It,
we could play it. And we do have a lot of questions about the calls. But
okay.
before we get to the singing, I want to ask a general question that we have, which is, what is the difference between Orthoptera and an orthopod? An arthropod, sorry.
Okay, arthropod is a very, very broad category for basically animals that have hard exoskeletons and jointed bodies. So it includes crabs, it includes insects, it includes spiders. Very, very broad category. Orthoptera is one order within the class Insecta. So it's like Orthoptera is at the same taxonomic level as Diptera, which are the flies, or Coleoptera, which are the beetles. And then, of course, nested under those orders, you have the families and subfamilies. King Philip can only fry German sausages. You remember that from your biology class. Kingdom, phylum, class, order, family, genus, species.
Terrific. Thank you. The book that you told us about, Morse's 1920 Manual, is that one of the ones that's now available if we all decide we need to go looking for that?
Yeah, I think if you Google it, you will probably, because I bought mine off the web, and in Google Books, which is where I came across it originally, there is a PDF of the actual, photographs of the page of the actual original version from 1920. I find the hard copy is easier to work with because you can page through it more easily, and I'm just kind of an old-fashioned guy. But a great, great resource, and there is, as you know, a long tradition of very high-level natural history work by amateurs going back into the 19th century. And there is a wealth of information compiled by our predecessors, and more of that is becoming available and making it possible now to put time series together, say that this is what occurred in Massachusetts in 1890 or 1940, and here is what occurs here now, and we can see the changes. So powerful, powerful tool.
Neat. Now, talking about the changes, you showed us where the populations were expanding, moving north, maybe moving to populated areas. You talked about some areas where you knew the populations had been there for a long time.
Mhm.
But so much in nature now is at risk and is declining. Is that true of the Orthoptera too, or are they holding on pretty steadily?
It's true. The same thing is true of Orthoptera that is true of most other things, that some species are doing very, very well. I mentioned Melanopolis femur rubrum, the red-legged grasshopper.
Mhm.
It is everywhere in Massachusetts. But if you read through Morse's species accounts over and over and over again, he says generally distributed in southern New England, and it's a species that I've never seen. You know,
Okay.
I think a lot of that is general habitat fragmentation. It's also probably the fact that in the 18th and 19th century, much of New England was deforested for farming, and Orthoptera are primarily species of open habitats. And so as the region has reforested over the last 80 to 100 years, a lot of good Orthoptera habitat has been lost. And to some extent, Morse may have been experiencing an artificially caused bounty of Orthoptera in New England.
Fascinating. We have a question about talking about what's happening to it versus other creatures in our ecosystem. We have a question about its role in the ecosystem and its role in the food web. How?
Great question. Like most kinds of insects, they are prey for a lot of things. Sparrows, savanna sparrow, grasshopper sparrow got its name because of its penchant for eating grasshopper nymphs. Spiders catch a lot of Orthoptera because as they hop they don't see the web, and the next thing they know, they're getting eaten by a spider. So they're in the food chain. They are often serving as prey items for a wide range of other arthropods and vertebrates. In terms of what Orthoptera eat, they do reach densities where they are influential in the plant ecology of a site. If you've got a density of, say, 30 grasshoppers per square meter, and they prefer to eat one plant in preference to all those other plants, you will see a decline in the prevalence of that plant. So they shape plant communities subtly sometimes and less subtly some other times. So like any other animal, they have a role to play, and they are both on the consuming and the being consumed end of the stick.
Neat. I have more questions about how to find them, but there's a great interest being shown here in their calls. So we're going to talk about their calls for a little while first. We have one question. Since Barbara's so good doing all our technical work, we're going to ask Barbara's first. She says, can one tell the temperature from all Orthoptera that sing as one can with a cricket?
Not precisely. And really, it is only one species of cricket that the frequency of the call is precise enough so you can use it as a thermometer. That's one of the tree crickets. But they definitely slow down in cold weather. I mean, you need to think of an Orthoptera call as having multiple layers. There is, in the case of a cricket, you've got chirp, 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 chirp. But if you think back to the slide I showed of the visual representations, within each of those chirps, you have pulsations. And so sometimes the pulsations will slow down. You know, if you think about plucking a comb, can you hear that? Maybe not.
Yep, no, we can hear it.
Or okay, so it like slows down, like, because that's basically what they're doing, is they're rubbing a comb together. Right? It's a reticulated piece of their wings, or they're rubbing a thigh against the side of their body. So they do slow down in cold weather, and towards the end of October and into November, you will be hearing these poor, pitiful, last surviving individuals dragging their calls out most piteously. So there is a connection, but you can't really precisely tell the temperature in most cases.
Okay, that was another one of our questions. When in the fall or winter do the crickets stop singing?
It depends on where you are. A hard frost will take care of most of them. Here on the vineyard, where we have an ocean moderated climate and our fall lasts longer than on the mainland, I will get crickets into late November, early December.
Wow.
And I've had grasshoppers in my yard, in adult grasshoppers left over from the fall in early January. So they're able, for the most part, they're able to withstand a light frost, but not a heavy one, and they're pretty good at finding warm microclimates on a cold night. So they will persist fairly late into the season, but they're already senescing. Go ahead.
And then the grasshoppers that you saw in February that you said come out at times.
Yeah, though that's a different case. Those are overwintering, and so they're at the start of their lives. But they will come out on a day when if it's sunny, all they need is the upper 30s or lower 40s, and they'll be out bopping around, and you can find them like on a south-facing fence line or something, where there is a little warm microclimate.
Oh, neat. We have another sound question, which is, I thought the Katydids were called that because that's what their calls sounded like, but apparently not. So why are they called Katydids?
Okay, the problem with, you notice that I use scientific names almost exclusively through this presentation, and that question is a really good example of why I do that. Katydid is a generic term. We have about 23 species of katydids on the vineyard. One of them, the true katydid, common true katydid, and I'm blanking on the scientific name, but it's there, there is one, is the one that I mentioned going cha-cha-cha, cha-cha-cha, or Katydid, Katydid, Katydid. Other kinds of katydids have long trilled calls. Other kinds of katydids have short explosive ones that go slick. If you've ever heard a Henslow's sparrow, some of them sound kind of like a Henslow's sparrow. Um, there is a katydid that says something like katydid, and that katydid has given its name to the entire class of these green Orthoptera with long antennae that we know collectively as katydids. So that's a really good question, and I'm really glad somebody asked it. Common names are helpful, but they're often misleading, and a lot of times they aren't well standardized. So if you use a common name, you can confuse people very, very badly. Birds have common names that are really well standardized. Butterflies do, dragonflies kind of do. Orthoptera, not so much. Flies, forget it. So if you're going to study insects, you have to get used to using the scientific names.
So how do you talk about these calls and you have such wonderful examples? How do you capture that noise? You said one time you used your iPhone and it wasn't so strong. What do you usually use?
The cell phone works pretty well. You just use I have the voice recorder application. I don't actually have an iPhone. I have an Android device, but I've had pretty good luck just using the voice recorder application that came with the phone. And you can edit it and cut it in its size. And you can actually, if you're using iNaturalist, you don't need to use photographs for observations. You can actually upload an audio file into iNaturalist. And so that works very well. I also have a little handheld digital audio recorder that produces much more high quality audio files. But it's another piece of equipment to carry around, and since I always have my cell phone, I usually leave the digital audio recorder at home and just use the cell phone. Give it a try. If you can get close to a katydid or a chorus of katydids, just turn it on, recording, and see what you come up with.
That's really neat. Between your instructions and what we learned from John Regeson, who I think joined us tonight, in his iNaturalist presentation, we're going to be having a lot of fun.
You can use your cell phone for bird calls also.
Yeah, that I'm used to doing. This is going to be fun to try. You talked about individual calls, and what is that? Why do we have any sense of what it is? Is it with some birds that have sort of their own dialects? What's
good question. I suppose that in the case of a species that, and some Orthoptera are pretty solitary, so you don't often hear more than one at a time.
Okay.
But there are ones that call in choruses. The common true katydid is one example, and Hapithus saltator is another example that I showed you and played the call for. And you do get individual variation in the call, and I'm not sure if there is a reason for that or if it just is, but it may very well be the case that even if it just is, it has an effect on the reproductive success of individuals, that females generally, or one particular female, may prefer one guy's call to another guy's call. And so then that would, if it were all the females that had a preference for the higher pitched, say, rather than the lower pitched, over time it would be the higher pitched ones that were able to reproduce more successfully, and that would influence the evolution and the genetic composition of that species going forward. So this is PhD stuff. There is a dissertation in there that some intrepid graduate student can take on.
That sounds like fun. Okay, how are we doing on time here? We've got time for a couple more questions. How to choose. One question we have is what adaptations do Orthoptera have to evade predators besides coloration? Or is it simply that, like schools of fish or flocks of birds, that they have safety in numbers?
They rarely are close enough together so that the schooling effect would have much benefit for them. Um,
Okay.
but they are cryptically colored. We looked at the example, for example, of the seaside grasshopper that blends in with its substrate, and Hepithus saltator, which is not so much cryptically colored, but it sings from the underside of branches, so it's really, really hard to find. I mentioned that while calling meadow katydids like to be a foot or 18 inches off the ground on a bunchgrass stem, that probably allows their song to radiate over a broad radius. It doesn't get muted by vegetation, so their chances of attracting a female are better. But behaviorally, as soon as they get wind of something wrong, like you approaching or a bird flying overhead, they let go and they drop nose first down into the base of the bunchgrass clump, and they dig in and they disappear. So they're cryptically colored. They blend in with their background. They're very good at keeping themselves concealed. And when they uh, are disturbed or feel threatened, they're even better at getting concealed. They'll burrow down deeper, and they're very, very hard to find. They're also pretty tough insects, particularly the larger katydids. It takes a serious bird to kill one of those bush katydids. I think they fight back. They can kick pretty hard. They can bite pretty hard, and they don't go down without a fight. So.
Well, seeing you hold them, that I found that amazing. That they've got to be pretty sturdy for you to be able to hold them like that and then take off again, right?
Yeah, I handle them gently. I'm ashamed to say that once in a while I break a leg off or something, but they are pretty tough. I will say that even our biggest species, they can give you a bite that you can feel, but they can't break skin or do any serious damage. If I had a live one, I'd let it chew on me for a while to show you that.
Oh,
Um,
oh,
but if you handle them gently with the wing species, I usually try to catch them by the wing
okay.
and hold them by the wings. You can also catch them in your hands like this and just sort of get a gentle grip on them by a leg or sometimes by the body. And as I mentioned, in a number of cases, if you're going to get a photograph of the diagnostic characteristics, you pretty much need to get them in hand. So it's a skill to work on. I actually enjoy handling them, and it's really funny because most of them, they panic when you first grab them, but if you're still, once they've calmed down a little bit, they forget that they're supposed to be frightened. And you may have noticed that I had some that were just sitting on my hand or sitting on a
Yeah.
finger. They'll stay there because they like your warmth. They'll stay there for a while, and you can photograph them at your leisure.
Oh, that's amazing. Well, let's end with my asking you for a tip for we novices, because the way I usually find them is I'm walking through grasslands with my grandsons running up ahead. They scare them, they go jumping away, and then I sort of try to follow after them. So if we are in a calmer environment and searching, how do we start to look for them?
Well, your grandson has exactly the right idea. They are very, very difficult. Most insects are very, very difficult to spot when they're sitting still. Um, and why not? Everything wants to eat them. There are predators all over the place. Being visible is a really stupid thing to do if you're an insect. So they're all masters of concealment. And the way I go insect hunting is to walk through habitat slowly, keeping my eyes down on the ground and wait for things to fly or hop or whatever they do, mark where they land and approach that spot more slowly and then look for them. With Orthoptera that call, you can also try to zero in on them, although that's surprisingly difficult to do. But I'll give you one little tip. When crickets and katydids are calling, they often like to wave their long antennae like fly rods, like they're fly fishing. And when you are looking at a plant and trying to find the katydid, the antennae are different from any of the plant parts. And often, surprisingly often, it is those fine thread-like antennae that are the first part of a calling katydid that you actually find. So that's a little pro tip that you can. But yeah, the way to find them is pretty much to disturb them. You can also do things like sweep netting. if you're really
Okay.
serious about it.
Yeah.
But if
Okay.
you're interested in just observing and photographing them, it's just a matter of bumping them into action and finding and watching where they land and then approaching them more stealthily. The grandchild approach.
Okay, I like that. Thank you so much. Now we can experiment on our own until we can somehow find a way for you to take us on a field trip.
Oh,
But we'll,
I would love to sometime. I don't make it to the mainland often. Why would you if you live on the vineyard? But
exactly,
maybe we can make it
exactly.
happen sometime.
We'll come to you. We'll work on that.
Yeah.
Next board meeting, everyone. Okay, well, thank you. This has just been absolutely amazing. If you have questions, viewers, you may submit them to Matt at mpelikan@biodiversityworksmv.org, or if it's easier for you to remember, through webinars at newtonconservators.org, and we will make sure that they get to him. Most of all, thank you, Matt, so very much, and hope to see everyone soon. Good night. Bye-bye.